Welcome to the Deep Dive, the place where we turn massive textbooks into a clear, compelling conversation.
And today we are taking on, well, a pretty monumental task.
We really are. We're diving into the core principles of animal life, a section you'd probably find in a textbook as Unit 7, Animal Form and Function.
That's right. And if you've ever wondered how an organism actually works, not just its parts, but what they do, this is the conversation for you.
We're basically asking the big questions. How does an organism stay alive, move, fight infection, and reproduce in a world that is, constantly changing?
Exactly. The focus here is on that inseparable link between structure, which is the anatomy, and the physiological processes, which is the function. It's about how form and action are tied together by evolution.
So our mission today is to take what is, I think, 11 incredibly dense chapters of biology and, well, distill them.
Right, into a coherent narrative, something you can actually follow and visualize.
Exactly. We want to cut through the jargon so you can picture these mechanisms, like how a single molecule can, make a muscle twitch, or why a reptile's waist looks the way it does. We want these ideas to stick. Okay, so where do we even begin with something this huge?
We have to start with the most fundamental truth in all of this, which is that form follows function.
Okay.
Natural selection basically ensures that an animal's anatomy, its structure, is perfectly matched to its physiological needs. And surprisingly often, this is all dictated by, well, basic physical laws.
The laws of physics, that's
Yeah.
fascinating, especially when you talk about something like size.
Right. As an animal gets bigger, its volume increases much, much faster than its surface area.
The old square cube law.
The very same. And that has huge implications. It means the skeleton, whether it's internal like ours or an external one, has to get disproportionately thicker just to support the weight.
And that thickness eventually limits how fast you can move.
It absolutely does.
And there's a perfect, almost famous example of this, right? The whole debate around the T-Rex.
Oh, absolutely. The Tyrannosaurus Rex, we all have this image of it, thundering along.
Right, chasing the Jeep.
Exactly. But physics might have something to say about that. We know it was massive, maybe 8, even 10 tons.
So the calculations on how fast it could actually run,
Mm.
they're all over the place.
They really are. Some models, if you just look at muscle force, suggest it could hit maybe 30 kilometers an hour.
Wow, like an Olympic sprinter?
Pretty much. But, and this is the big but, other researchers look at the sheer stress on the skeleton.
And say it would have just snapped its legs.
Precisely. The argument is it might have been more of a very fast walker. The sheer mass puts a hard cap on its mobility.
Okay, so size is constrained, but function also demands that every single cell in the body gets what it needs.
Right. Every cell needs access to an aqueous environment for exchange. Nutrients in, waste out.
For simple organisms, that's easy.
Super easy. Take a hydra. Its body is just a 2-cell layer thick sac. The outside cells are in pond water. The inside cells are in fluid. Done.
Or a tapeworm.
A tapeworm is perfect. It's so flat. It has this huge surface area, so it just absorbs everything it needs directly from its host. Simple diffusion does all the work.
But for us, for a seal, for any big, complex animal, diffusion from the outside is just not going to cut it. So how did evolution solve that?
By folding. It got really creative with geometry.
So it's all about internal surface area.
Exactly. Think about the alveoli in your lungs or the villi in your intestine. There are all these extensively folded or branched surfaces. If you could somehow flatten out all the internal exchange surfaces in your body.
I've heard this statistic. It's huge, right?
It's more than 25 times the surface area of your skin. This massive internal folding is the only way a complex animal can meet the demands of trillions of cells.
And when you see unrelated animals facing the same problem, they often evolve the same solution.
Convergent evolution. That's one of my favorite concepts.
The classic example is fast swimmers.
It's the best one. You take a tuna, which is a fish, a penguin, a bird, and a seal, a mammal.
Totally different evolutionary paths.
Hundreds of millions of years apart. Yet they all have that same torpedo shape. Why? Because that shape minimizes drag in the water. The same physical problem leads to the same structural solution. Form follows function.
Okay, so to really understand the function, we need to look at the hierarchy of the structure. Cells make tissues, tissues make organs, and so on.
Right. And we can basically categorize every animal structure into just 4 main tissue types.
First up, the boundaries.
Epithelial tissue. These are just tightly packed sheets of cells. They're the barriers, the interfaces. They cover your skin, line your gut, form your glands.
They're the gatekeepers.
Right.
So how do they stop a pathogen from just squeezing between the cells.
That's where tight junctions come in. These are protein structures that literally seal the space between cells, making that sheet of tissue really robust and selective about what gets through.
Okay, so you have the lining,
Right.
then you need the stuff that holds it all together.
And that would be connective tissue. And it's sort of the opposite of epithelial tissue. Instead of packed cells, you have sparse cells scattered throughout this non-living extracellular matrix.
And that matrix is the key part.
It's the secret sauce. It can be a liquid, a jelly, or a solid. The cells in there, like fibroblasts, are the ones that actually secrete the protein fibers for the matrix. And you've also got defense cells, like macrophages, patrolling around in there.
And this tissue type includes some really specialized things like bone.
Bone is a perfect example of mineralized connective tissue. You have cells called osteoblasts that lay down a collagen matrix, and then it gets hardened with calcium and phosphate.
And has that very specific structure.
In hard bone, yes.
Yeah.
These repeating units called osteons, they look like little bullseyes around a central canal that holds nerves and blood vessels.
And on the complete other end of the spectrum from bone, you have blood.
Which is the only liquid connective tissue. Its matrix is the plasma, and you have cells suspended in it. Erythrocytes for oxygen, leukocytes for defense, and platelets for clotting. It just shows the incredible range of one tissue type.
So we have structure and protection. Now we need an engine.
Muscle tissue, defined by one thing, its ability to contract.
And we have three main types.
Right. Skeletal and cardiac muscle are both striated. They look striped. Skeletal is for voluntary movement. Cardiac is the heart. And then smooth muscle isn't striped, and it handles the slow, involuntary stuff like digestion.
And finally, the wiring that controls it all.
Nervous tissue. This is for receiving, processing, and sending information. The key player here is the neuron.
With its dendrites and axons.
Exactly. Dendrites receive signals, axons transmit them, and they're all supported by helper cells called glial cells.
Okay, so you have all these specialized parts. They have to be coordinated perfectly, and animals have two major systems for that communication.
The high-speed nervous system versus the slower broadcast endocrine system.
The nervous system is like a dedicated telephone line.
Exactly. It's wired, it's fast, it's specific. An electrical signal goes right to a precise location for an immediate response.
Whereas the endocrine system is more like a radio station.
Perfect analogy. It broadcasts hormones into the blood. They go everywhere. It's slower, but the effects can be really long-lasting. And critically, a hormone only affects cells that have the right receptor for it.
And both of these systems work together to maintain that stable internal environment, homeostasis.
Right. And when you think about homeostasis, it's useful to think about two main strategies. You have regulators and conformers.
Okay, what's the difference?
A regulator, like a river otter, uses a ton of energy to keep its internal state, like its body temperature, constant, even if the outside world is changing wildly.
So the otter is warm even in freezing water.
Exactly. A conformer, on the other hand, like a largemouth bass, just lets its internal temperature match the water around it. It conforms to the environment.
And temperature is probably the variable we talk about most. Let's start by getting rid of the term cold-blooded.
Please. It's so inaccurate. The real terms are endothermy and ectothermy.
Endotherms make their own heat.
Right, birds and mammals. We generate most of our heat internally through metabolism. Ectotherms, like lizards, get most of their heat from the outside world.
But that doesn't mean they're cold.
Not at all. A lizard basking in the sun can easily have a body temperature that's warmer than ours. It's about the source of the heat, not the temperature itself.
So one of the key ways animals manage their temperature is by controlling blood flow to the skin.
Absolutely. Vasoconstriction is when surface blood vessels narrow. That shunts warm blood away from the skin and keeps you from losing heat.
And vasodilation is the opposite.
Right. You widen those vessels, rush blood to the surface, and dump heat when you need to cool down.
But the most ingenious adaptation for dealing with cold has to be the countercurrent heat exchanger.
It's a masterpiece of biological engineering, a perfect example of form solving a functional problem.
So think of a goose standing on ice.
Exactly. Or a dolphin's flipper in cold water. You can't afford to lose all your core body heat through that extremity. So the artery carrying warm blood out to the foot is placed right next to the vein carrying cold blood back.
And the heat just transfers from the warm artery to the cold vein all the way down.
Precisely. By the time the arterial blood gets to the foot, it's already cool, so it doesn't lose much heat to the ice. And the returning venous blood has been pre-warmed, so the body doesn't have to work as hard to heat it back up. It's incredibly efficient.
And we even see some pretty amazing temperature control And animals we usually think of as ectotherms, like in that Burmese python experiment.
That study was fantastic. The female python coils around her eggs. Researchers found that as they lowered the temperature in the chamber, the python started these rhythmic muscle contractions, basically shivering.
And they measured her oxygen consumption.
And it shot up, which is a direct measure of metabolic rate. So she, an ectotherm, was actively generating metabolic heat specifically to keep her eggs warm. It shows you how blurry these lines can get.
Okay, so all this function, from shivering pythons to maybe fast-walking T-Rexes, it all requires fuel. And that brings us to metabolic rate.
Right, the total energy an animal uses over time. We measure in kilocalories, the calories you see on a food label, or more often by measuring oxygen consumption.
Because oxygen is the key ingredient for burning that fuel.
Exactly. You can even stick a shark in a tank and measure its oxygen use to figure out the metabolic cost of swimming.
Now, let's get back to the paradox of size, that weird $1.75 power relationship. What does that actually mean for, say, a shrew versus an elephant?
It means the shrew is living life in the metabolic fast lane, 247.
Okay.
Because of that scaling, a tiny animal has a vastly higher energy consumption per gram than a huge one. The shrew has to eat pretty much its own body weight in food every single day just to stay warm.
Whereas the elephant's cost per gram is much, much lower.
Way lower. It's a much more efficient machine in that sense.
So when animals face an energy shortage, what are their strategies? They can't just keep burning fuel at that rate.
No, they have to go into conservation mode. Torpor is a short-term version, like a daily dip in metabolism and body temperature.
And hibernation is the long-term version.
Right, the seasonal deep dive version. And hibernation is just incredible. I mean, in some hamster studies, their body temperature drops to near freezing, metabolism slows to a crawl, and get this, their internal biological clock, the SCN, actually stops ticking.
It just pauses time.
It literally suspends their 24-hour cycle until they warm back up.
So when they're not in suspended animation, animals need a constant supply of raw materials. Fuel, yes, but also what we call essential nutrients.
Right, these are things the body needs but can't make itself. Certain amino acids, certain fatty acids, vitamins, minerals.
And with vitamins, there's a really important distinction to make.
A critical one. The water-soluble ones, like vitamin C and B, if you take too much, your body just flushes them out.
But the fat-soluble ones are different.
Very different. Vitamins A, D, E, and K can actually build up in your body fat to toxic levels if you overdo it. So, balance is key.
Okay, so once you eat the food, it goes through 4 stages.
Ingestion, which is eating. Digestion, breaking it down with enzymes. Absorption, taking up the small molecules. And finally, elimination of whatever's left over.
Let's follow a piece of food through the human system, starting in the stomach. It's a pretty hostile place.
It's an engineering marvel. The gastric juice has a pH of about 2, super acidic.
From hydrochloric acid.
Right. And that acid does 2 things. It kills most of the bacteria on your food, and it denatures proteins, causing them to unfold. Then the enzyme pepsin can get to work, and pepsin is special because it works best in that extreme acid.
Most of the actual absorption happens later, in the small intestine.
That's the main event. The small intestine has this ridiculously huge surface area, thanks to all those folds, villi and microvilli.
The tennis court inside your abdomen.
That's the one. It's how you maximize the space you have for absorption.
And getting the most important nutrients across the intestinal wall isn't a passive process.
Not at all. Simple stuff like fructose can diffuse across, but for essential amino acids and glucose, the body uses active transport. It spends energy to pull those molecules in, even against a concentration gradient.
To make sure you get every last bit.
Every last bit. It's about maximizing efficiency.
And you can often tell what an animal eats just by looking at the length of its digestive tract.
Absolutely. Carnivores have shorter guts because meat is pretty easy to break down. Herbivores need much longer, more complex systems to deal with tough plant matter.
Like the koala.
The koala is a perfect example. It has this incredibly long gut to slowly ferment and extract nutrients from those tough, low-protein eucalyptus leaves.
And the most specialized plant eaters of all are the ruminants.
Your cattle, sheep, deer, they have that 4-chambered stomach. The first 2 parts are basically huge fermentation vats full of mutualistic microbes that can break down cellulose.
And they chew their cud.
They regurgitate it, rechew it to increase the surface area, and swallow it again to give the microbes another shot at it. It's a whole process.
Okay, once the nutrients are absorbed, the body has to manage its fuel reserves. What's the order of operations for burning fuel?
First, it taps the easy access stuff. Glycogen in the liver to maintain blood sugar. then glycogen in the muscles to fuel the muscles themselves.
After that's gone.
It turns to fat in the adipose tissue. Fat is the most energy-dense storage you have. It can keep you going for weeks.
But the moment-to-moment regulation is all about blood glucose, keeping it in that really tight window.
Yes, that's glucose homeostasis, and it's a classic negative feedback loop run by the pancreas.
So after a meal, blood sugar goes up.
And the beta cells in the pancreas release insulin. Insulin tells your cells to take up glucose and your liver to store it.
And when blood sugar drops.
The alpha cells release glucagon, which tells the liver to break down that stored glycogen and release glucose back into the blood. It's this constant push and pull.
And when that system fails, you get diabetes.
Right. And we need to distinguish between the two main types. Type 1 is an autoimmune disease where the body destroys its own insulin-producing cells.
So they need to take insulin.
Exactly. Type 2 is different. That's when the body's cells stop responding properly to insulin. It's called insulin resistance, and it's strongly linked to lifestyle and is becoming a massive global health problem.
And beyond just blood sugar, our body regulates long-term energy stores by controlling our appetite with hormones.
It's a whole conversation between your gut and your brain. You have stimulants like ghrelin, the I'm hungry hormone from the stomach.
And then the suppressants.
Right, PYY from the intestine after a meal, insulin. And the big one for long-term stores, leptin, which is released by your fat cells.
And the discovery of leptin came from one of the most clever, if a little gruesome, experiments in modern biology.
The parabiosis experiment. Absolutely foundational science.
So they took these obese mice with different genetic mutations.
Right. One mouse, the off mouse, couldn't make the satiety hormone, leptin. The other, the DB mouse, made leptin but didn't have the receptor for it. So the experiment was to surgically connect their circulatory systems to see what would happen.
Okay, so what happened when they connected a normal mouse to the one that couldn't make leptin?
The normal mouse's leptin circulated into the ob mouse, suppressed its appetite, and the ob mouse lost weight. That proved the hormone existed.
But the really wild part was when they connected a normal mouse to the DB mouse, the one without the receptor.
Oh, that was the kicker. The DB mouse stayed obese, of course, but the normal wild-type mouse, it starved to death.
Why?
Because the DB mouse, with no receptor, was pumping out massive amounts of useless leptin. This flooded the normal mouse's system and completely shut down its appetite. It proved not only that the hormone existed, but that the receptor was just as critical. Just brilliant.
Okay, moving from getting energy to distributing it. In any big animal, you need a circulatory system.
Diffusion is just way too slow over more than a few cell widths. You need plumbing.
And there are two main designs.
The open circulatory system, which you see in insects and other arthropods, the heart pumps a fluid called hemolymph, but it just sort of bathes the organs directly in open spaces.
It's lower pressure, less energy intensive.
Right. And they can use it for other things, like how spiders use that fluid pressure to extend their legs.
But for higher performance, you need a closed system.
The closed circulatory system. Blood is always contained in vessels. This allows for much higher pressure, which means faster and more efficient delivery of oxygen and nutrients. It's essential for active animals like us, or, a squid.
And for mammals and birds, with our crazy high metabolic rates, we need double circulation. Why is that separation so important?
Because mixing oxygen-rich and oxygen-poor blood is incredibly inefficient. Double circulation creates two separate loops.
The pulmonary and the systemic.
Right. The pulmonary circuit sends oxygen-poor blood from the right side of the heart only to the lungs. The systemic circuit then takes that freshly oxygenated blood from the left side and pumps it at high pressure to the rest of the body. That separation is key to our high metabolism.
Okay, let's walk through that path in the mammalian heart, the four chambers.
So blood comes into the two atria, which are the receiving chambers. They push it into the two powerful muscular ventricles, which are the main pumps.
And the left ventricle is the strongest.
By far. It has to pump blood to the entire body. The right ventricle only has to pump it next door to the lungs. And you have 4 valves that act like one-way doors to make sure the blood never flows backward.
What's amazing is that the heart beats on its own. It doesn't need the brain to tell it to contract.
It's autorhythmic. The signal starts right in the heart muscle itself, in a spot called the SA node. That's the master pacemaker. It sets the rhythm. The nervous system can speed it up or slow it down, but the basic beat comes from the heart.
And that pumping action is what creates blood pressure.
Right. The peak pressure during contraction is your systolic pressure. The pressure that remains when the ventricles relax, thanks to the elastic arteries, is the diastolic.
Now, blood velocity changes as it moves through the body. It slows way down in the capillaries. Why is that?
It's all about total cross-sectional area. A single capillary is tiny, but you have millions of them in parallel. So the total area is huge, much bigger than the aorta.
Like a river widening into a lake.
Perfect analogy.
Hmm.
The flow slows to a crawl, and that's critical because it gives time for all that exchange of nutrients and waste to happen.
And the body is constantly redirecting that flow based on need.
Yeah, through vasoconstriction and vasodilation.
Yeah.
By narrowing or widening the small arterioles, the body can shunt blood away from, say, your gut and toward your muscles when you start exercising. It's incredibly precise.
We've talked about the fluid inside the vessels, but some of it always leaks out. That's where the lymphatic system comes in, right?
The unsung hero of circulation. The lymphatic system is basically a drainage network that collects all the fluid and protein that leaks out of the capillaries. Without it, you'd swell up with a condition called edema.
And the lymph nodes are part of that.
They're the filters. They're packed with white blood cells. So as the lymph fluid passes through, they trap and kill any pathogens. That's why your glands swell up when you're sick. The lymph nodes are working overtime.
Okay, inside the blood itself, the key players for oxygen are the red blood cells.
The erythrocytes. Each one is packed with about 250 million molecules of hemoglobin. It's an incredible packaging system that increases the blood's oxygen capacity about 100-fold.
But a tiny change to that hemoglobin molecule can have devastating effects, like in sickle cell disease.
It's a textbook example of how a single amino acid change can alter function. The altered hemoglobin forms rigid fibers when oxygen is low, which distorts the red blood cell into that sickle shape.
And those sickled cells cause blockages.
They clog up small blood vessels, which is incredibly painful and causes organ damage. They're also much more fragile and don't live as long, which leads to anemia.
Another major circulatory problem is the hardening of the arteries.
Atherosclerosis. It starts with inflammation in an artery wall. Then you get this buildup of lipids and cholesterol and other gunk, which forms a plaque.
And that narrows the artery.
It narrows the artery, restricts blood flow, and makes it much more likely for a blood clot, a thrombus, to form. If that clot blocks an artery in the heart or brain, that's a heart attack or a stroke.
Which is why interventions like stents are so important.
Exactly. A stent is basically a little mesh tube that's inserted to prop the artery open and restore that blood flow.
The whole point of circulation is to support gas exchange.
Yeah.
And the one physical principle that drives all of it is partial pressure.
That's it. Gases only move from a place of higher partial pressure to a place of lower partial pressure. That gradient is the only thing that matters.
And different animals have evolved amazing structures to maximize that gradient. For fish, it's their gills and countercurrent exchange.
We saw countercurrent for heat, and it's the same principle here for gas. In fish gills, water flows over the gills in the direction opposite to the blood flowing through them.
And that's the key, the opposite direction.
It's everything. If they flowed in the same direction, the pressures would equalize about halfway through and exchange would stop. But because they flow opposite, a steep gradient is maintained along the entire length of the gill.
So the blood is always encountering water with a slightly higher oxygen pressure.
Always. And it allows them to pull up to 80% of the oxygen out of the water. It's incredibly efficient.
Insects, on the other hand, just sidestep the whole problem.
They really did. Their tracheal system is this network of air tubes that branch throughout their body and deliver air directly to the cells. Their circulatory system doesn't even carry oxygen.
Which is why they can have such a high metabolism for things like flight.
Right, the flight muscles themselves help pump air through the system, getting all the oxygen they need.
Now for us, what controls our breathing rate? It's surprising that it isn't a lack of oxygen.
It really is. The main driver is the level of CO2 in our blood. When you exercise, you produce more CO2, which dissolves in your blood and makes it slightly more acidic.
And the brain detects that change in pH.
The medulla oblongata, specifically, it senses that drop in pH and immediately triggers faster, deeper breathing to blow off that excess CO2 and bring the pH back to normal.
And even hemoglobin itself is dynamic. It changes how tightly it holds on to oxygen based on local conditions. That's the Bohr shift.
The Bohr shift is so elegant. In your active tissues, where cells are burning fuel and making lots of CO2, the local pH drops. That lower pH actually changes hemoglobin's shape and decreases its affinity for oxygen.
So it lets go of more oxygen right where it's needed most.
Exactly. It's a perfect self-regulating delivery system.
We can't leave this without mentioning the absolute champions of oxygen management, the diving mammals.
The Weddell seals, the Cuvier's beaked whales. They're physiological marvels. They stockpile O2 in their blood and muscles, and then they ration it. They shut down blood flow to non-essential organs, slow their heart rate way down.
To make that one breath last for an hour or more.
An hour, sometimes 2 hours, and they can dive nearly 2 miles deep. They operate at a level of oxygen efficiency that is just, well, unimaginable for us.
Okay, let's move on to defense. Survival means having a robust immune system, and we have two main branches.
The first is our innate defense. This is the non-specific rapid response team.
So things like skin, mucus, stomach acid.
Exactly, physical and chemical barriers. And then you have the cellular defense, primarily phagocytosis.
Where immune cells literally eat invaders.
Right. A macrophage or a neutrophil will engulf a bacterium into a little vacuole, fuse it with a lysosome full of destructive enzymes, and just digest it.
And when you get a cut, the local response is inflammation.
Right. Damaged cells release chemicals like histamine. This makes local blood vessels dilate and get leaky.
Which is why it gets red and swollen.
Exactly. It's a signal to flood the area with fluid and defense cells to clean up the mess and fight off any invaders.
The other arm of the immune system is adaptive immunity. This one is slower, but it's specific, and it has memory.
This is the domain of the lymphocytes, the B cells and T cells.
B cells make antibodies.
Right, they recognize whole antigens floating around. T cells are different. They only recognize little pieces of an antigen that are presented to them on the surface of another cell by proteins called MHC molecules.
And the key to this whole system is clonal selection.
Clonal selection is how you get a massive targeted response. When a new pathogen shows up, only the one B or T cell that happens to have the perfect receptor for it gets selected to divide.
And it makes a huge army of clones.
A huge army. Some become the active plasma cells that fight the current infection, but crucially, some become long-lived memory cells. And those memory cells are why the next time you see that same pathogen, your response is so much faster and stronger. It's the whole basis for how vaccination works.
Now, in today's world, our adaptive immunity is facing a huge challenge from superbugs.
Multi-drug-resistant organisms, it's a massive crisis. Bacteria are evolving resistance to our antibiotics faster than we can develop new ones.
Which brings up that incredible story about phage therapy.
It's a fascinating revival of a forgotten idea. Bacteriophages are viruses that only infect and kill bacteria. They were used 100 years ago, but then we discovered antibiotics and sort of forgot about them in the West.
But with superbugs, they're making a comeback.
They are. The story of the patient who was saved after being infected with a bug resistant to dozens of antibiotics is just incredible. They had to hunt down specific stages that could target his particular infection, and it worked. It's this idea of using targeted biology to fight biology, and it's a very promising tool in this fight.
Okay, moving from external threats to internal balance. All animals have to manage their water and salt levels. That's osmoregulation.
And it's a constant battle. Some marine animals are osmoconformers. Their insides just match the stable ocean environment. But most of us are osmoregulators, actively spending energy to maintain a specific internal state.
For land animals, the biggest problem is just drying out.
Desiccation. They have to find water, conserve water, and even use the metabolic water that's a byproduct of cellular respiration.
And how you manage water is directly tied to how you get rid of nitrogenous waste. There's a big trade-off here, isn't there?
A huge one. It's a balance between toxicity, the energy it costs to make, and how much water you lose.
So option one is ammonia.
Ammonia, super toxic, so you need a ton of water to dilute it. But it's cheap to make energetically, so it's great if you're a fish living in water.
Option two is what we do, urea.
Urea, it's the compromise. Moderately toxic, needs a moderate amount of water, costs a moderate amount of energy to produce.
And option three is uric acid.
Uric acid, this is the water conservation special. It's not very toxic and takes almost no water to get rid of. It's a paste, but it costs a lot of energy to make.
So it's perfect for birds and reptiles.
Especially ones that lay shelled eggs. You can't have toxic watery waste building up inside an eggshell, so a solid, non-toxic waste product is a brilliant evolutionary solution.
And the organ that manages all of this in humans is the kidney, with its millions of tiny filters, the nephrons.
And the nephron's job has 4 key steps. First, filtration. Blood pressure just forces all the small stuff out of the blood into the tubule. It's not selective at all.
Which means you filter out good stuff too.
A lot of good stuff, which is why step 2, reabsorption, is so critical. This is where the body selectively uses active transport to pull back all the useful things. Glucose, amino acids, most of the water.
Then there's secretion.
Secretion is like targeted disposal. The body actively pumps specific wastes and toxins from the blood directly into the filtrate. And the last step is excretion, getting rid of the final product, urine.
And our ability to make really concentrated urine to save water is all thanks to the loop of Henle.
The loop of Henle is another countercurrent system, but this time it's for salt. It actively pumps salt out into the tissue of the kidney's medulla, creating this incredibly steep osmotic gradient.
So the deeper you go into the kidney, the saltier it gets.
Exactly, and that salty gradient is the key.
Because later, as the urine passes through that salty area in the collecting duct,
All that saltiness just pulls water out of the urine by osmosis, concentrating it and saving a huge amount of water for the body.
and this whole process is controlled by hormones.
Primarily ADH, antidiuretic hormone. If you get dehydrated, your blood gets saltier. Your brain senses this and releases ADH.
And ADH does what?
It goes to the collecting duct and tells the cells to insert special water channels called aquaporins into their membranes. This makes the duct super permeable to water, so you reabsorb as much as possible, and your urine becomes very concentrated.
So these control systems, nervous and endocrine, are running everything. Let's look a little closer at how they work, starting with hormones. Their chemical nature really dictates how they act, right?
It determines everything. You have your water-soluble hormones like polypeptides. Insulin is a good example.
They can't cross the cell membrane.
So they have to bind to a receptor on the outside of the cell. That triggers a chain reaction inside, often using a second messenger like cyclic AMP to get the job done.
And then you have the steroid hormones.
The steroids, like cortisol, they're lipid-soluble, so they just slip right through the cell membrane and bind to a receptor inside the cell. That hormone receptor complex then goes right to the DNA and acts as a switch to turn genes on or off.
And this communication often happens in cascades.
A whole chain of command. The hypothalamus tells the anterior pituitary what to do. The pituitary then releases a hormone that tells a target gland, like the thyroid, what to do. It's a way to amplify the signal and have multiple points of control.
And for a really fast response, like fight or flight, you get a combo of both systems.
It's the perfect combination. The nervous system gives a direct signal to the adrenal medulla, which then dumps epinephrine and adrenaline into the blood.
And what's so clever is how one hormone can do different things in different places.
It's all about the receptors. Epinephrine might bind to one type of receptor on blood vessels in your gut, causing them to constrict, but bind to a different type in your skeletal muscles, causing them to dilate. It's how you shunt resources to where they're needed most for escaping danger.
Okay, let's switch to the nervous system's language. The action potential. That all or none electrical pulse, how does it work?
It's all about voltage-gated ion channels. A stimulus pushes the neuron's membrane to a threshold voltage.
That's the point of no return.
Exactly. At threshold, voltage-gated sodium channels fly open. Sodium ions rush into the cell, making it rapidly positive inside. That's the spike.
Then how does it come back down?
Two things happen almost instantly. The sodium channels slam shut and inactivate, and voltage-gated potassium channels open. Potassium rushes out, taking positive charge with it, and the membrane potential drops back down. It's an incredibly fast, self-propagating wave.
So that electrical signal travels to the end of the neuron, but it can't jump the gap to the next cell.
Right. It has to be converted into a chemical signal. The action potential triggers the release of neurotransmitters into that tiny gap, the synapse.
Like acetylcholine.
Acetylcholine is a classic. At the neuromuscular junction, it's an excitatory signal that tells your muscles to contract. But in the heart, the same molecule is inhibitory. It binds to a different receptor that slows your heart rate down.
The specificity of that synapse is why drugs and toxins are so powerful.
It's a very sensitive pathway. Nicotine, for example, is a stimulant because it mimics acetylcholine at some of its receptors. And at the other extreme, you have Botox,
Botulinum toxin.
the most potent toxin known. It works by blocking the release of acetylcholine altogether. No acetylcholine means no muscle contraction, which is why it's so dangerous, but also why it works cosmetically to temporarily paralyze facial muscles.
Looking at the bigger picture, we divide the nervous system into the central and the peripheral. And within the peripheral, you have the autonomic system controlling all the involuntary stuff.
And the autonomic system is this perfect balancing act. It has two divisions that are constantly in opposition.
The gas and the brake.
Exactly. The sympathetic division is the gas pedal. It handles fight or flight, ramps up your heart rate, burns energy. The parasympathetic division is the brake. It handles rest and digest, slows things down, conserves energy.
Inside the brain itself, specific regions handle specific jobs.
The hypothalamus is the master homeostatic controller. Temperature, hunger, thirst, the endocrine system, it runs it all. The thalamus is like the brain's central sorting office for sensory information.
It decides where all the signals go.
It does, for everything except smell. It sends the signals to the right parts of the cerebral cortex, the brain's outer layer where the complex thinking happens.
And if you map that cortex, you see that the amount of brain space dedicated to a body part isn't about its size.
Not at all. It's proportional to the density of sensory nerves or the degree of fine motor control. The hands and the face have these huge representations in the sensory and motor cortexes compared to, say, your back.
The cortex also has specialized centers for language.
Two really critical ones. Broca's area is for producing speech. Wernicke's area is for understanding language.
So damage to Broca's area you can understand, but you can't speak fluently.
Right, damage to Wernicke's, and you can speak fluently, but it's nonsensical because you can't comprehend language properly.
And our sense of time, our daily cycle, that's also controlled by a specific brain region.
The suprachiasmatic nucleus, or SCN, in the hypothalamus, it's the master biological clock, and it gets synchronized to the 24-hour day by light coming in from your eyes.
And we know it's the clock because of that hamster experiment.
The transplant experiment, they took the SCN from a mutant hamster with a 20-hour cycle and put it into a normal hamster with a 24-hour cycle. And the normal hamster started living on a 20-hour day. It proved the SCN is the pacemaker.
And when these circuits fail, the results are devastating, like in Alzheimer's or Parkinson's.
It shows how fragile it all is. Memory is based on neuronal plasticity, the constant remodeling of synapses. Alzheimer's disrupts this with tangles of tau protein. Parkinson's is caused by the death of just one specific group of dopamine-producing neurons. The loss of a tiny part can have catastrophic consequences for the whole system.
Okay, let's turn to the ultimate measure of evolutionary success: reproduction.
And animals have evolved a huge diversity of strategies to get it done.
Including some asexual ones, like parthenogenesis.
Parthenogenesis is when an egg develops without being fertilized. Male honeybees are a great example. They develop from unfertilized eggs.
And hermaphroditism.
Right, having both male and female systems. It's really common in animals where it might be hard to find a mate, like a barnacle stuck to a rock. It doubles your chances.
In mammals, though, it's all about the precise hormonal synchronization of the female cycle.
The ovarian cycle, which is about the egg maturing, and the uterine cycle, the buildup of the uterine lining, have to be perfectly coordinated.
And the key hormones are estradiol and progesterone.
Right. As the follicle with the egg matures, it pumps out more and more estradiol. When estradiol hits a certain peak,
It triggers the LH surge.
exactly. That spike in luteinizing hormone is the direct trigger for ovulation. Then the leftover follicle turns into the corpus luteum and starts making progesterone, which maintains the uterine lining for a potential pregnancy.
And if pregnancy occurs, you need the placenta.
The placenta is the amazing life support system. It's the interface for nutrient, gas, and waste exchange. And it has to act as an immune barrier because the fetus is genetically foreign to the mother.
And birth itself is driven by a positive feedback loop.
A classic one. Uterine contractions cause the release of the hormone oxytocin. Oxytocin causes stronger contractions, which causes more oxytocin release, and the cycle escalates until the baby is born.
But before any of that can happen, you need fertilization. And the egg has to prevent polyspermy, right? More than one sperm getting in.
It has a multi-layered defense system. First, the sperm's acrosomal reaction releases enzymes to digest the outer jelly coat.
Then the blocks go up.
Instantly. The fast block is an electrical change in the egg's membrane that repels other sperm. And that's immediately followed by the cortical reaction, which hardens the outer layer into the permanent slow block.
Once that one sperm is in, the embryo starts a massive reorganization called gastrulation.
Gastrulation is when the simple ball of cells rearranges itself into the 3 primary germ layers, which sets the fate for every cell in the body.
So you have the ectoderm, mesoderm, and endoderm.
Ectoderm is the outer layer. It becomes skin and the nervous system. Mesoderm is the middle layer, muscle, skeleton, circulatory system. And endoderm is the inner layer, which becomes the lining of the gut and associated organs.
And this process is driven by cell-to-cell communication. One group of cells telling another what to become.
That's induction. A group of cells releases chemical signals that induce their neighbors to change their fate.
And this was proven in that legendary experiment with the frog embryo.
Speeman's organizer experiment. They took the tissue from the dorsal lip of one embryo, the organizer, and grafted it onto the belly of another embryo.
And what happened?
The host's own cells were induced to form a second, complete head-to-tail embryo on its belly. It was definitive proof that specific groups of cells act as organizers, directing the entire body plan.
And we see that same principle, organizing complex structures like our limbs.
A developing limb bud has organizing centers like the ZPA, the zone of polarizing activity. It secretes a protein called sonic hedgehog that patterns the limb along the thumb to pinky axis. It's all about signals and gradients creating complex 3D structures.
All right, to wrap things up, let's talk about how animals interact with their world. Sensory input, motor output, and behavior.
This is where it all comes together. And senses like touch and hearing rely on mechanoreceptors.
In hearing, you have to convert sound waves in the air into an electrical signal. How did that final step happen?
Some ways make your eardrum vibrate, which moves the middle ear bones, which creates waves in the fluid of your snail-shaped cochlea. Those fluid waves bend tiny hair cells on the basilar membrane. And that physical bending is what triggers the nerve impulse.
So a mechanical force opens an ion channel.
Precisely. And balanced works similarly, but with gravity. Little dense particles called otoliths in your inner ear settle with gravity and bend hair cells, telling your brain which way's up.
Vision, on the other hand, is chemical.
Right. Light hits pigments in the rods and cones of your retina. And here's the weird counterintuitive part.
Okay.
In the dark, your photoreceptors are constantly releasing an inhibitory neurotransmitter. When light hits them, it causes them to hyperpolarize, which stops them from releasing that inhibitor.
So stopping the stop signal is what sends the go signal to the brain.
Exactly. It's a double negative that creates a positive signal.
Okay, from sensing to doing. The engine of all movement is the sarcomere in our muscles.
It's the basic contractile unit.
Yeah.
And it works by the sliding filament model. You have your thick myosin filaments and your thin actin filaments.
The myosin heads are the motors.
They are. They grab onto the actin, pull, let go, and grab again, powered by ATP. This pulls the actin filaments toward the center. The filaments themselves don't shorten, they just slide past each other.
And the switch that allows this to happen is calcium.
It all comes down to calcium. A nerve signal triggers the release of calcium from the sarcoplasmic reticulum. The calcium binds to a protein called troponin, which then pulls another protein, trypomyosin, out of the way.
Uncovering the binding sites on the actin.
And that allows the myosin heads to grab on and start the contraction. No calcium, no contraction.
When we look at the energy cost of moving, there are huge differences.
Big differences. Swimming is the most efficient form of locomotion because the water supports your weight. Running is the least efficient, especially for small animals.
But the kangaroo has a neat trick.
The kangaroo is super efficient at hopping because its tendons act like giant rubber bands. They store energy when it lands and release it on the next hop, which saves a ton of metabolic energy.
And finally, the ultimate expression of all this machinery, behavior. And we analyze it through two lenses.
Proximate versus ultimate causes. Proximate is the how, the immediate mechanism. Ultimate is the why, the evolutionary advantage.
And behavior can be learned, like imprinting in geese.
Right, that time-sensitive learning. Or spatial learning, which is about creating a cognitive map of your environment.
That's the Digger-Wasp experiment.
The classic. The wasp used a ring of pine cones to find its nest. When researchers moved the pine cones, the wasp searched for its nest in the middle of the empty ring, not where the nest actually was. It was following its internal map, not the nest itself.
And then there's social learning, learning from others.
The vervet monkeys are amazing at this. They have different alarm calls for leopards, eagles, and snakes. And the young ones have to learn not only which call means what, but the correct evasive action for each one. It's a life or death lesson.
When it comes to finding food, it's all about optimal foraging.
Balancing the energy you get from food against the risk of getting eaten yourself.
Like the mule deer.
Exactly. They were observed choosing to eat in open areas with lower quality food over forest edges with better food. Why? Because the risk of being ambushed by a mountain lion was way lower in the open. Survival trumps calories.
And when animals compete, you can get these complex dynamics, like in the side-blotched lizards.
It's a real-life game of rock, paper, scissors. You have three types of males. The aggressive orange-throated males beat the territorial blue-throated males. The blue-throats beat the sneaky female-mimicking yellow-throats, and the yellow-throats beat the orange ones by sneaking past their defenses.
So no one strategy is always the best.
Which is why all three persist in the population. It's a constant cycle.
And that brings us to the final puzzle. altruism, seemingly selfless behavior.
Which, from a genetic perspective, is often actually selfish. The explanation is kin selection. You help your relatives because they share your genes.
And this is quantified by Hamilton's rule.
Right. Hamilton's rule just says that an altruistic act is favored by natural selection if the benefit to the relative, multiplied by how related you are, is greater than the cost to yourself.
And the naked mole rats are the ultimate example of this.
The whole colony is so highly related that the non-reproductive workers will literally sacrifice themselves to protect the queen and kings because that's the best way to ensure the survival of genes that are almost identical to their own.
And with that, we have come through an absolutely massive journey. From the physics of a T-Rex to the genetics of a mole rat. We've seen how structure and function are linked, how bodies are fueled, defended, and regulated with incredible precision.
We have. And if there's one single idea that ties all of this together, for me, it's that life is maintained not by being static, but by a constant, dynamic balancing of opposing forces.
A managed conflict.
That's the perfect way to put it. It's insulin versus glucagon, vasoconstriction versus vasodilation, the sympathetic versus the parasympathetic nervous system, even the aggressive lizard versus the sneaky one. It's all about these opposing forces reaching a temporary functional equilibrium.
So really, stability comes from a constant state of flux.
Exactly. So the next time you just stand up or your mood shifts or your heart rate changes, I want you to think about that. Think about the thousands of tiny molecular conflicts that are resolving themselves into a perfect working balance inside you in that single second. The animal body isn't calm. It's chaos managed into perfect functionality.
A perfect chaotic machine. What a fantastic place to end.
Yeah.
Thank you for joining us on this incredibly deep dive into the form and function of the animal world. Keep exploring.